genetic and biochemical studies have revealed that three E. coli proteins, namely MutS, MutL, and MutH, are dedicated to mismatch repair. Although these proteins are essential for mismatch repair, they are not sufficient. Several additional enzymes and protein factors also make important contributions towards this mismatch repair. Among these enzymes and protein factors are DNA helicase 2 or UVRD, single stranded DNA binding protein SSB, 5' to 3' exonucleases, EXO7 or RECJ. The fourth one is 3' to 5' exonucleases, that is EXO1, EXO7 or EXO10. The fifth one is DNA polymerase 3 holoenzyme. The next is DNA ligase, and finally deoxyadenosine methylase. So there are a number of enzymes and protein factors which are involved in this mismatch repair system in E. coli. The process of repair begins when MutS, which is a homodimer or a homotetramer, binds to the mismatch. So first step is the binding of MutS protein to the mismatch. MutS recruits MutL, a homodimer, in an ATP dependent fashion. So when this MutS binds with the mismatch, it also binds MutL to this site. So a complex is formed. Then the MutS MutL complex activates MutH, which is third protein, which makes an incision at the nearest unmethylated GATC site, either 5' or 3' to the mismatch in the newly synthesized DNA strand. So in the first step, MutS is attached, then MutL is attached, and when this complex is formed, this complex activates MutH to introduce NICs either on the 3' side or 5' side. So two NICs are introduced by the MutH protein. So here you can see this is the site, damaged site, mismatch site, and this is MutS. It binds to the damaged site and then it recruits MutL. This blue one is MutL, and this complex is formed. This complex is MutS MutL DNA complex, and this complex activates the third protein MutH, which introduces NICs, and two NICs are introduced on the either side of DNA strand. So in this way mismatch repair takes place. MutH shares sequence homology with the type 2 restriction endonuclease SAU3A1. Both enzymes recognize and cleave GATC sequences. MutH doesn't bind or cleave fully methylated GATC sites, whereas SAU3A1 cleaves fully hemi and unmethylated GATC sites. So this is difference between the MutH and SAU3A1. Next in the process is that helicase 2, which is also called UVRD, unwinds the DNA, and SSB protein binds to the resulting single strands. So next is the unwinding at the mismatch site, and so that SSB may bind to the unwound strands. When the incision is 5 prime to the mismatch. So if mismatch is here and the incision is towards 5 prime, exo 7 or rect J exonucleases hydrolyze the nicked strand in a 5 to 3 prime direction. However, when the incision is 3 prime to the mismatch, that is on this side, exo 1, exo 7 or exo 10 exonucleases hydrolyze the nicked strand in a 3 to 5 prime direction. So two nicks either on 3 prime side or 5 prime side may be introduced. So different exonucleases then cleave these nicks. DNA polymerase 3 holoenzyme fills the gap with new DNA. DNA ligase seals the remaining nick, and deoxyadenosine methylase adds a methyl group to the GATC site. So in this way, this mismatch repair is complete. So here you can see this is the mismatch site. So here you can see a MutS MutL complex. This complex activates MutH, and this MutH introduces a nick here. So this nick is towards the 3 prime side, and similarly it may be towards the 5 prime side. So the next is the helicase 2, SSB and ATP. They take part in the pathway, and this portion or this portion, then these are hydrolyzed, and now this is the gap, and this gap is filled by DNA polymerase 3 holoenzyme, and in this way a complete strand is formed with no mismatches. All organisms that have a mismatch repair system have MutS and MutL homologs. However, MutH is present only in E. coli and some other gram-negative bacteria.